Morning, everybody. Still got a frog in my throat. Oh, hello.
Oh, sorry, Professor. I just wanted to ask you a quick question.
Go for it.
I had a little visitor here, and I was wondering if you wanted to see my dog.
Oh, of course. I'd love to see your dog.
Okay, let's see. Is she sleeping? Come here, Evie. This is Evie. She likes to
Evie.
fall down next to me when I do class, and this is her.
Oh,
Yeah.
it's hard to know one end from the other, to be honest. She's so fluffy.
Right? Evie, look.
Oh, that's my youngest daughter's name, Evie.
Really?
Mm-hmm.
Well, I wanted to show you because the other one didn't want to come in, but she won't leave me alone.
Oh, yeah, I'll show you one of mine. That is Pepper.
Hi.
She was actually a puppy when I was teaching online first time around, which is funny.
Really?
Yeah, she used to Zoom bomb my classes all the time.
Well,
Yeah.
she's gonna learn about genetics today.
Yeah, she's a little cutie pie. Okay, let's just check where we're at. But thank you for showing me Evie. She's a cutie pie.
Of course.
All right. I think that'll do. Let's open up the other one. We're going to be working off of PowerPoints today. So 1 and 2, if you want to open those for yourself. That's what I'm just doing myself at the moment. Fresh in my memory. Yeah, there's nothing much interest in number 1. I'm not even sure why we have that one. Okay, so let's get cracking, as they say. Dum dum dum. So let me share my screen with you. Get rid of that cough. Bring up chat. Share screen. All right, where are we at? Here we go. Okay, so typically I pitch the beginning of the class or course rather as being a bit of a recap session, essentially. Largely because, actually, let's get this up. I want to write out what we're going to be doing this section. Hello, Michael. Largely because, you know, some of you have only just taken Gen Bio 1 and similar. Some of you haven't taken it for years and years and years, like if you've just come back to college. So it doesn't hurt to have a bit of a kind of refresher, essentially. So this is going to be section 1, right? So as I said, there's 4 sections. They kind of walk our way through genetics in a kind of somewhat meandering fashion. Frog in my throat. So this is what I do at the beginning of every section there. And typically what I do at the beginning of every class, period 2, right? Obviously it's a lot easier when I have a bigger whiteboard in front of me. So essentially what we're going to be doing this section for the next, what is it, month or so, give or take? I don't know, recap basic genetics. Going to be introducing a lot of terminology, a lot of words, right? This is something that I stressed throughout the course. We have a particular language that we use. And so if you want to be able to understand what I'm talking about, be it in class or in exams or whatever, you need to know the language, right? And so that's something that is worth studying kind of in and of itself, right? You're learning a language. So what do we do next? So basic genetics, essentially structure of DNA, recap of gene expression and the like. What's next? Should have looked up the schedule, shouldn't I? Mendelian inheritance. Ooh, that's a stretch. And math. I'm sorry to say there's a bit of math in genetics. Can't get away from it, unfortunately. It's not my strong suit either. But we're going to be, in this section at least, this is mostly probability, right? Which is pretty intrinsic part of Mendelian inheritance, right? It's all about probabilities of possible outcomes, right? And basically being able to test whether those outcomes are what we expect or not, right? Or close to what we expect, right? So that's a big part of the math of this section. There'll be some math in the next section too. We'll get to that when we get to that. All right. Then there's non-Mendelian inheritance. I'm not sure if Mendelian is 1 or 2 ounces. Let's give it 2. Right. And in my mind, this is the interesting part. Of Mendelian inheritance, right? Because although this Mendelian inheritance is kind of fun, it's like a bunch of logic games, basically, right? Where you kind of try and figure out what you're going to get with particular combinations of alleles and genotypes and the like. It doesn't really explain very much human genetics, right? What makes you who you are, right? And in many ways, that's kind of the most interesting part of genetics, at least in the beginning. Whereas non-Mendelian inheritance really explains pretty much all of it. Not entirely, right? There are a few congenital diseases, so ones that you have with you when you're born. Oh, up, Luna. Up. And my other dog came in. Well, one of my other dogs. Things like cystic fibrosis, sickle cell disease, phenylketonuria, you know, stuff like that, right? Are Mendelian traits. Everything else is non-Mendelian. How tall you are, whether you're plump or skinny, whether you're brown or white, whether you have diabetes or not, right? All rests on non-Mendelian inheritance. Polygenic traits, incomplete dominance, which I have new examples of, which is super neat, and various other things, right? That's non-Mendelian. Oh, there's cricket. Hey, come up. That's cricket, long-nosed pest. She actually surpassed herself this week, and she smells very nice as a result, because she got a bottle of air freshener from my daughter's room. Yes, you. Hello. I think she's got a long nose, hence the name long-nosed pest, and punctured it. So, like the whole top floor of the house stinks of air freshener. My wife almost passed out in her office, which is where it happened, because it's such a strong smell of air freshener. And even now, this is several days past, cricket still smells of air freshener. Very odd as well. Good smelling dog. And then to finish up, we're going to cover cell division, cell cycle, mitosis. This is a stretch. Meiosis. Handwriting is even worse than usual. All right, so that's basically the outline of what we're going to be doing. We should get through most of this today, I think. Having a quick look back through the PowerPoint, there's not a whole bunch more that would take us past one lecture. And then we'll start on Mendelian inheritance next week in person. And the cool thing about Mendelian inheritance, so you probably know all about Mendel's P's, which in my view are boring as batshit. So we're going to be, I'm just going to do a we're going to be using cats, because cats are way more interesting and they have lots of really cool genetics to talk about. Anyway, so that's what we're going to be doing. And sorry, dog. Oh, here's Luna. Hey, Luna. Come here. I've got all three with me now. That's Luna. She's the original dog, the OD. Aren't you? So now I have three dogs. First for me ever, having three dogs while teaching. There you go, online. Let's just go off. There's not enough space. All right, let's get back to material at hand. All right, so basically, what I want to do today is just go over some of the basics. We'll be hitting this stuff in one form or other multiple times over the course of the semester. But one thing that I like to do is to introduce concepts or terminology, like ideas, right? And then come back and, oh yeah, sure, give me a sec. And then come back and hit them again and again, right? So it's the whole kind of idea of iteration. There we go. You got that, Joshua? I'll be posting up a picture to Blackboard later anyway, in case you miss anything. Need any longer? No? All right, awesome. Let's get back. Oh, right. Okay, well, anyway, as I said, these pictures go up to Blackboard, so you'll catch them the second time around if you missed anything first time. Okay, and so obviously what underlies everything in genetics and all biology really is DNA, at least for almost all, yeah, I'd say all organisms. I don't actually consider viruses an organism. Doesn't really meet all the criteria in my mind. Some people do. So obviously some viruses use RNA to store their information. COVID is one of those, flu, influenza, HIV, et cetera, et cetera. And actually that's kind of a fun, interesting fact is that RNA sucks in terms of a storage molecule because, give me a sec, Viviana, because it's so error prone, right? So its replication is hugely error prone. That's why we have so many variants of COVID, you know, different versions of flu every flu season and the like, simply because they accumulate way more mutations than DNA viruses do. Question about, oh, we are currently on PowerPoint 2, which is basically kind of the where we properly get into the material of the intro stuff. The PowerPoint 1 is just a, you know, some terms and things. In terms of the textbook, I couldn't tell you, to be honest. Maybe, I guess so. You'd have to check yourself. Okay, so there's, again, I don't have enough space on this computer screen. It's only a very small screen. There's some really important stuff involved in this basic structure. Let me get some. I know there are like fancy tools and stuff on Zoom now, but I'm a bit of an old-fashioned Luddite and I haven't actually figured out how to use it. So all of this, there we go. No, we want no fill. There we go. Right. This is the basic structure of DNA, right? One strand of DNA, right? Because DNA is a double-stranded molecule, which is a a super important part of its properties, right? So this structure, I'm going to get rid of this and use, annotate. Now let's go a little play with this. All right, so, look at that. See, an old dog can learn new tricks, so to speak. Right, that there is one basic building block, right? So this is a nucleotide. I'm going to type stuff in the chat, right? Because it's kind of a good way of keeping notes, essentially, right? And this consists of 3 parts, right? Sound like I'm dying here. I'm actually really quite feeling good, but still got a frog in my throat. So a nucleotide consists of 3 things, right? We've got the phosphate, spell it, right? And this is negatively charged. We'll come back to this several times over the semester. There's a 5-carbon sugar called a ribose. That's where the ribonucleic acid, or the ribo part of that name, comes from. Sorry, guys. There we go, see if that helps. And there's the nitrogenous base. Right, and the nitrogenous base, that's the basically that A, C, T, and sorry, could get my punctuation straight. There you go, that'll do. Right, and the nitrogenous base is hang on, there we go, this part here, right? And so you'll see that for these 3, thiamine's not present for whatever reason. They're different in shape. There we go, now I got it. And this is central to their properties, right? And they're essentially their identity, right? Because the nitrogenous base is the only thing that differs from one nucleotide to another, right? And this is where the information that encodes the proteins that actually do stuff, right? Because DNA is just a store of information. It's not something that does anything itself, right? It has no other properties, right? All of that information is stored, well, almost all that information. I have to be careful with sweeping statements in biology because they don't apply all the time. Is stored in these bases. Some other stuff is stored in modifications to DNA and modifications to protein that wraps DNA around themselves. But for the large part, information is stored in these, right? And so what you'll see is there are, let's see if I can draw little arrows. All right, this is like drawing in paint, right? There are multiple hydrogens attached to oxygens and nitrogens. That's where the nitrogenous bit comes from. And what you'll see, I think I've got it right. I have. As they say, even a blind squirrel finds a nut every so often, right? There you go. God, that is absolutely awful bit of drawing. But anyway, hopefully you could actually get something from that. Anyway, what you'll see is there are different numbers, different positions, different shapes, right? And so all of that is super, super important because essentially these groups, right, they are polar groups, right? Oops, capitals. So the stuff highlighted with arrows badly are polar groups. Here's a bit of a recap for you. Going to ask questions so you're not just sitting there listening to me talk all the time. What kinds of bonds do polar groups form? See if anybody remembers. So these have polar, what's the name of the word? Something bond. Can't remember. That's a long time since I taught Gen Bio 1. But they have polar bonds, right, which are slightly different in charge from one end to the other. So these dipole, maybe. I don't actually know what that is, to be honest with you. Covalent, no. Actually, that was the word I was scratching around for, Alfred. Thank you. So they are polar covalent bonds, right? So that's what these lines here. Don't know if you can, like that line there, and so on, right? Those are covalent bonds. These ones in particular are polar covalent bonds, right? So they're slightly charged, right? They're not ions, right? Because there's no missing what's-its here and there, right? Let's bring my, can you all see my cursor? I'm pointing at stuff. Want to know that? Oh, you can? Good. Great, because I don't want to point at stuff and you're not be able to see it. Right, so these particular, right, so these aren't, these atoms aren't missing, right? So these are not ionic bonds. They're different type bonds. So they're not covalent, right? Things that form bonds with this. They're not ionic. They're not van der Waals, because no one can remember, right? What's the other type of bond that you can form? Fairly weak. Yeah, thank you, Joshua. Hydrogen bonds, and whoever else was about to speak. That was probably Michael. Hydrogen bonds, right? And so these little bits sticking out the side are what are responsible for the formation of hydrogen bonds, right? And so hydrogen bonds are what hold the two strands of DNA together. Can anybody remember any of properties of hydrogen bonds? Kind of gave them already if you're listening, but it doesn't hurt to stress this. Where do hydrogen bonds sit in the I don't know, panoply of bonds in terms of strength? Are they the weakest? Are they the strongest? Which ones are the strongest, actually? Can anybody remember? They're not quite the weakest, almost. They're weak, but not the weakest. Great, exactly. Van der Waals are the weakest ones. Covalents are strongest. Ionics kind of pretty strong. Water isn't around. And hydrogen bonds are kind of weak, but their number makes up for it, right? And so this is a really interesting property of hydrogen bonds, right? So they're relatively easy to break, right? Every time you boil a kettle or boil a pot of water, or even actually look at a disappearing puddle of water outside in the Texan sun, right? Oh, hang on, Joshua. Hydrogen bonds are not polar. They are the result of polar bonds in two different molecules. All right, so I might actually need to draw this. That'll be fun. Give me a second though. Right, so hydrogen bonds break relatively easily. That's why water basically kind of falls apart when you heat it to, you know, I don't know, probably even like 70, 80 degrees, but certainly 100 degrees Celsius when it boils, right? Those hydrogen bonds break and water molecules literally fly all over the place, and that's what steam is, right? So if you want to hold two strands of DNA together, right, but also be able to pull them apart, because you have to separate DNA to be able to copy it, or actually to be able to copy it, whether you're making new DNA or whether you're making RNA, you have to pull it apart. Right, so hydrogen bonds are basically like a zipper or Velcro, right, whichever you want to think, right? So hydrogen bonds are weak, but strong when there are lots of them. So basically lots of weak bonds added together, which is really good because it's basically something, it's like a series of weak magnets. You know, like those fridge magnets, which kind of suck. They don't hold much stuff on the fridge, right? They're like that, right? So they can be pulled apart, but when you let go of them, they'll snap back together again. And these number and position and shape are what dictate how many of those hydrogen bonds form, right? Just before I get into that, I'll let's put up a new, come back to this. That's weird. That will kind of, pardon me. All right. So before I get into the next bit, I have to remind myself what that is in a bit. Let's go through something that was it Joshua? Yeah, Joshua brought up. Right. So covalent bonds are sharing of electrons. So if you have carbon, oh, this is going to be painful. All right, I'm not going to do all of the bonds. Right. Basically, there's, you can try doing this with a trackpad. It's really hard. All right. So if you have a carbon and a hydrogen, carbon doesn't pull on electrons super hard. So it shares its electron with hydrogen's electron fairly equally, right? So they're shared, they're kind of like in the same orbit, as best I understand it, but they're shared equally, right? So if you're looking at kind of probability wise, you have about equal probability of finding it anywhere in here, right? So that's why we draw them in the middle. So this is called a, oops. All right, so if you thought my writing on whiteboard was bad, this will make it look better. There you go. That's a non-polar covalent bond. There should be a hyphen there. Now, when you have atoms that are really like hold on to their electrons, right? Enough that they will oxidize other atoms, strip electrons from them, right? Oxidize, what does that come from? It comes from oxygen, right? And so oxygen and nitrogen and also fluorine. Yeah, I think that's a atom. Don't talk about it much. Have a very strong pull. They've got a lot of protons in a relatively small size, and protons are positively charged and pull those electrons to them. So when you have the same deal, I'm just going to draw one because otherwise it'll be torturous, right? They're still sharing electrons, right? They're not ions, right? They haven't stripped electrons. But let's brighten this a little bit. But what they do is that most of the time they're pulling electrons closer to them, right? They're avaricious of electrons, let's say. And so that covalent bond, because it's still a covalent bond, is drawn like so, right? And essentially what that means is that, trying to do a dashed line, not very successfully. There you go. That's not that bad. I'm not going to try and draw sigma though, right? And so this area is slightly negative. It's not negative in the sense of chlorine, right? Which is an ion, right? It is slightly negative. And as a result, this area here, this actually isn't all that bad. Not quite as embarrassing as I thought it would be. That area is slightly positive, right? So this is called a polar There you go. Covalent bond. Now, if I'm to get rid of this, it's kind of fun. There we go. And draw up another proton. Oh, actually, no, we don't want that there. This has turned into a little bit of a tangent, but it's fine. Right. If we have, let's say, this, an oxygen carbon double bond. I think that's what's called a carbonyl group. Oh, Pepper's getting mad about something. Right. She's a small and fierce dog. She kind of really fits the kind of small, fierce dog trope. All right. Anyway, so now we have a slightly negative area, and basically we then get a hydrogen bond between them. And hydrogen bonds are drawn with these funky dashed lines, like so. You'll see the point of all this tangent in a little bit. It's an important point to make. Right, so that is, I'm just going to call it an H bond because otherwise we'll be here all day with you watching me scrawl like a child on Blackboard. Anyway, that's a hydrogen bond, right? So it's not polar in and of itself. It's the result of two polar bonds coming together, one slightly negative area, right, which is going to be a nitrogen or an oxygen typically, right? And one slightly positive area, which is going to be a hydrogen that's attached to a nitrogen or an oxygen, right? That's what gives you a hydrogen bond. That makes sense? I don't know if this is going to be saved. It's print screen. Cool. Awesome. It's actually really important because hydrogen bonds are something that are at the root of an awful lot of biology. Where are we at again? All right, here we go. So essentially, coming back to this, right? The number of hydrogen bonds differs between the bases. Don't you remember which bases pair with which? So what is an adenine base pair with? If anybody can remember. There's a cute mnemonic that helps you remember this. I just can't remember what it is. Sorry. Yeah, thank you, Alfred. A's base pair with T's, right? So adenine, which is this one, right? Base pairs with thymine, right? And this one has, at least in, unfortunately, we don't have a thymine here to show. This one has two groups that can form hydrogen bonds. How many hydrogen bonds do you reckon are between A and T? Ah, Josh has already beat me to it. 2. Now, when you look at guanine, you'll see 3. All right. And there are 3, as Joshua said, 3 hydrogen bonds between guanines and cytosines. Right. So the shape and the number of hydrogen bonds that can form are part of DNA base pairing. right? Which is how we get accurate DNA replication, how we get accurate transcription into mRNA, and a whole bunch of other things as well. Okay, so in addition to that, what you'll see, this is, actually, let's clear this. This is a single ring, right? So cytosine and thymine are what are called pyrimidines. And those are single ring bases. And I'll tell you a mnemonic as well. Why, oh, why? And some of you have heard this before, it's kind of cheesy, but it works. Am I such a small base? That's how I remember it. So I imagine they're like, oh, why am I such a small, miserable little base when I have these big base brothers around me, sisters? All right. Whereas adenine and guanine are purines. Those are two-ring bases. And one thing you'll notice is if cytosine base pairs with guanine, 1 plus 2 get 3. Thymine is a single ring, adenine is a 2-ring, 1 plus 2 is 3, right? So the actual size of the bases is important too. Right, so you got size, you got shape, and you got the number of hydrogen bonds that can form. That is the essentially the property that underlies DNA, right? That's why DNA is a double-stranded molecule. All right, so enough on that. Kind of went on a little bit longer than intended. Next thing that is important is, actually, let's clear stuff. This business, right? So DNA is unidirectional. Actually, is that the best way of putting it? Well, let's use more words and be clearer. I guess unidirectional does work. But anyway, sometimes more words are better. All right, so what you'll see is that one end of DNA is different from the other, right? At this end, we have our phosphate group, right? And at this end, we have a 3' hydroxyl group, right? So OH is hydroxyl. And one of another super important points to make, so there's a lot of information here. I actually really like this image. There's a lot going on here, right? So that OH group is the only place a new nucleotide can be added. The only exclamation mark, right? So DNA can only be made in a 5' to 3' direction. Same goes for RNA, actually. You can't add new bases up here, right? Doesn't work. You can only add them at this end, down here, right? And so DNA DNA has direction, RNA has direction. The proteins that are made from not from RNA, using RNA as a template has direction. right? So all of this kind of comes together to kind of ta-da, make like this magic synthesis, right? And there are in between, as a result of that OH being used, there are phosphodiester bonds, right? These are the covalent bonds that link one phosphate group here to the next ribose sugar here, right? And that gives us the phosphoribose backbone. And that's alternating. Alternating phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, so on and so forth. All right. So this directionality is going to become really, really important when we talk about DNA replication and transcription, right? In fact, in DNA replication, it causes all kinds of problems. All right, so that's enough of that slide. Ooh, let's move on. Let's see where we're at next. Okay, covered that already. One thing to point out here, let's go back to annotation, is that DNA is double-stranded. Also, super important as well, it's anti-parallel, right? So you don't have both strands going in the same direction. You have one going in one direction, one going in the opposite direction. So if I were to be a pedant, which I often am, in this strand here, right, which is, oops, bollocks, sorry, shouldn't swear. My finger slipped on the trackpad, and my trackpad's not very big. All right, so this one here, if I were to be a pedant, those letters should be upside down. All right. So anti-parallel, another thing to point out is that, let's get rid of this bit. It's not an even twist, right? So it's not like you're getting a twist tie or a bit of rope or wire and just twisting them together like that, right? So DNA is not an evenly twisted molecule. It is a alternating molecule. So this is the, sorry, everybody, inflicting more terrible finger writing on you. This is the minor groove. This is the, which is kind of embarrassing, sorry, everybody, major groove. There we go. Hello, Alyssa. Right. And this is also super important because this is part of the shape of DNA, right? This is part of how proteins recognize DNA to bind to it. And an awful lot of the proteins that interact with DNA, transcription factors, regulatory factors, so on and so forth, do so by recognizing this shape, right? Actually, let's get rid of those, otherwise I'll copy through to the next page. All right, so we will talk, come on, there we go. We'll talk later about the structure of DNA. Oh crap, I think I forgot something on the board. Never mind. There is other stuff in section 1. I just ran out of space to put it there. Yeah, we'll talk more about the ultrastructure of DNA. So this is kind of like the bare bones version of DNA. It doesn't look like this in your cells, right? It actually is in a much more complex, organized, structured form called chromatin. Right. Okay, so Next, obviously, this doesn't do anything, right? It stores information only. Now we need to get this DNA, this information out, right? And that process is called gene expression. And gene expression, which is often called the central dogma, kind of irritatingly, it's my like one-man crusade against inappropriate use of that term. So gene expression is basically DNA, mRNA, protein. Right, there's a lot more to it than that, right? There are many slips twixt cup and lip, as Shakespeare would say. But that's the basic outline. And if I'm going to be super pedantic, non-coding genes are just DNA to RNA. There you go, things like ribosomal RNA and stuff like that. But either way, that information has to get out of that DNA into a form that can be accessed, right? So DNA is in the nucleus, proteins are made in the cytoplasm, right? So the intermediate is the mRNA, and then that has to get translated, right? So there, here we go. That's this kind of process there, here, right? Hey, see, I think I changed that. Actually, no, Joshua, non-coding genes are super interesting. We don't tend to talk about them a huge amount. We'll cover them a bit later on in the semester. So non-coding genes, basically those RNAs are produced and those RNAs do stuff. So they're either catalytic, so ribosomal RNA, so the things that actually make proteins or synthesize peptide bonds, those are actually RNA molecules, they're not proteins, even though ribosome is what's actually put down here. They can be structural, transfer RNAs, which are also used in translation. What else? SNRPs, small nuclear ribonuclear proteins, SNRMPs. So these are kind of both, right? So basically, long story short, non-coding RNAs are a whole separate group of things which do stuff, right? Their functions vary depending on what they are and what their purpose is. They're involved in controlling gene transcription, involved in translation, splicing, all kinds of things actually, but they do not encode proteins, right? That's why they're non-coding genes. So introns are a whole separate deal, right? Introns are bits of RNA which are not needed or wanted for translation. And you can interrupt as much as you like, I don't mind. That's the whole point, right? So we'll get into gene structure a little bit later on. So this is basically the flow of information, right? You have DNA, that DNA is copied, right? It's not turned into anything, it's copied in something, into mRNA. That mRNA is then exported, right, out of the nucleus where the DNA is, into the cytoplasm where the ribosomes are. And then that RNA is translated into protein, right? And something that also bears mentioning as well, right? Where's my, we have, that's supposed to be a 2. There you go. We have 2 copies of each gene, right? Man, I can't believe I'm spending so much time of a lecture finger writing, right? We've got 2 copies of each gene, right? We can have potentially hundreds or thousands of copies of mRNA from that gene. And you can just have lots of protein. And depending on how those mRNAs are treated, right, whether they're broken down or protected or whatever, right, whether translation is inhibited or promoted, so on and so forth, we'll get varying amounts of protein from those, right? So this isn't just a flow of information, it's also an amplification of information as well, right? And how that's controlled, right, that's really, quote Shakespeare again, I've never actually read Shakespeare. But anyway, therein lies the rub. That's where so much of what makes us who we are comes from, is not this, right? It's all of this, right? How that information is accessed, where, in response to what, right? So on and so forth. Obviously, there are differences between us all in terms of our protein sequences, but those differences are actually really quite small, right? Relatively speaking, in terms of number. Okay. So we, what is it? Probably the 3rd section. I'm thinking straight. 3rd section of the course is almost entirely focused on this process. The last section of the course is focused on how this process is controlled, which really is where the interesting stuff is, in my mind. Right? So in terms of transcription, right, and again, this is, for many of you, it's just going to be a recap. Oh, good question, Andres. So that's an interesting question because it's kind of a slightly kind of off an angle from where you need to be, I think. So mRNAs are always translated correctly, as far as I'm aware. I don't think there are really any errors in translation. I don't think. There may be, right? Because I'm not an expert at that. But what you're probably thinking of is, what if the sequence that is being translated results in an incorrect protein, right? I think that's probably what you're thinking of, right? If that's the case, it depends, right? It depends a great deal. And actually, we're going to do one of my trademark bouncing arounds. And so what you're thinking of are mutations, right? Changes in the sequence of a gene, right? Which alter potentially the sequence of a protein, maybe, right? Could also alter the control of transcription as well, right? So where's my pointing stick? So if that mutation happens to be in typically the 3rd base of a codon, and again, we'll get to what those things mean in a little bit. That mutation is what's called silent. Has no effect whatsoever, right? We have loads and loads and loads of those mutations throughout our genome, right? Throughout our coding genome, at least, right? Because it doesn't make any difference, right? So sometimes you'll get a mutation that results in a change in the protein sequence, right? These are called missense mutations, right? Which is what we've got here. That, it depends, right? Whether that has an effect or not. So a great example, and I like using myself as examples a lot. I don't respond to pain medication very much, right? So Ibuprofen doesn't do very much. The injections you get when you're having a tooth kind of pulled out or worked on doesn't do very much. And I've actually had to stop my dentist in the middle of pulling out wisdom teeth till I go, hurts, right? Because it really friggin' hurt. He didn't give me enough painkiller. He didn't believe me at the beginning, but he did after I kind of pulled on his leg until he gave me some more. Right, and that almost certainly is due to a change in the protein sequence of it wasn't a lot of fun, but I was kind of like spaced to the nines anyway, so I just thought it was funny. A protein sequence change in the receptor for that medication, right? Most likely, there are also other ways in which you can vary in your tolerance. Could also be that I have a protein that breaks down pain meds faster than other people do, right? So even though you can put a load into me, it gets metabolized really quickly and excreted into my urine, right? So those kind of small differences can have fairly large effects depending on where they're at and what protein they're in, and a whole bunch of other conditionals, right? Now, these ones down here, hang on, let's get rid of this. Sorry, this is time consuming. Right, so these mutations here, these result in the introduction of what's called a stop codon, either directly through a nonsense mutation, right? So that's the direct creation of a stop codon. And stop codons stop translation, right? So wherever a ribosome sees a stop codon, whoosh, off it goes. No more protein synthesis, right? Or you can change the frame that is being read, right? And we'll talk about reading frames and the like a lot more when we get to translation proper, right? But that typically results in the creation of a new stop codon, right? If actually, I do hope you are impressed by my finger painting. Don't worry, you don't all have to congratulate me at once. Sorry, I'm just having fun. They're catastrophic. They result in no functional protein being produced, right? And so those are what are called loss of function mutations, right? So you don't have a change in function, you just don't have function. So that is depending on what gene that occurs in. That could be something that I don't know, results in you having ginger hair, red hair, for example. That's result in some people loss of function of a melanin receptor, or it could result in you not existing at all, right? In terms of it being a lethal mutation, right? So context there is also super important as well. So there are lots of other ways to categorize mutations as well. Some of the more interesting ones are the ones which don't change the protein sequence at all. They change how the gene is regulated, right? And so these are, in my mind, the sort of mutations that have really driven a lot of evolution, particularly of us, right? So not mutations that change our proteins, but mutations that change essentially where and how much those proteins are expressed or produced, right? So there's also kind of like other different kind of terminology here it's worth going through this just because there's some important words in here. Here's another, oops, if I get this right. Important word, right? Germline mutations. So essentially, it doesn't matter how many mutations, actually, it doesn't matter how many mutations I accumulate now, right? Because I'm not going to be having any more kids for various reasons. 2 is like plenty enough for me. But also, even if I did get mutations in my arms and my legs or whatever, and I were to have more children, those mutations would not be passed on because they are not in my germline, right? So the only mutations that you will pass on to your children, if you're going to have any, are those which occur in your gonads, basically, right? In your sperm and your eggs, right? Any that occur in your somatic tissue, right, which is everything else, don't count. They obviously might be bad for you, right? Because, you know, skin cancer sucks, but it doesn't affect your offspring, right? So that's kind of some other words which are worth thinking about. Anyway, so that's kind of like the end of the beginning, but it depends. Mutations are very interesting, right? Because it really potentially changes everything or changes nothing, right? Anyway, so brief overview of transcription. Transcription is pretty straightforward. Simply what it is DNA is separated, which is what this diagram is showing you here. And one strand, called the template strand, is copied to generate a copy of the coding strand. Right, so in transcription, the template strand is used to make a copy of the coding strand. Right, and there's a whole bunch of stuff that goes into it. A lot of proteins are involved, right, to recognize where to do that, and then to actually make the mRNA itself. And in eukaryotes to process that mRNA, right, because it doesn't, the first version is not the final version, essentially. Prokaryotes are a lot simpler. But anyway, that then, where are we at? There's no, that one. That one. That then is used as a template and is read by a ribosome. I don't think we got a picture of a ribosome here. Let's see. Get this out of the way. You're probably somewhat familiar with this kind of images. I don't know why Beckman called it. That makes centrifuges. Anyway, that's a ribosome. So it's a protein complex that essentially feeds or assembles on and then feeds the mRNA through it and adds amino acids on in sequence, depending on the sequence of the mRNA, which is what you can see here. So now we come back to another bit of terminology, and that is not condone, codon. Codon is a group of 3 letters in mRNA. Doesn't matter which letters, right? There's just 3 of them, which equals 1 amino acid. Right, so essentially what you do in terms of translation is you take the 4-letter code of mRNA and you don't read it as individual letters, because if you did, you'd only have 4 amino acids, right? You don't read it as 2 letters, because if you did, then, oh, what would that be, like 8? Yeah, maybe. I suck at math. you eat it in 3, because that gives you more than enough, actually, possible combinations to encode all 20 amino acids. Right, so that's how you go from 4 letters of mRNA to 20 different amino acids. And that is what is basically encoded here, right? Oops. That's also why the genetic code is degenerate, right? It's not because it, likes hiding in the woods, drinking whiskey and playing the banjo or something like that. Not there's anything wrong with playing the banjo, but it's because more than one codon, 3-letter word, can code for the same amino acid, right? There are very few exceptions to that. There's only 2. Got to find them now. One is methionine, right? This is called the start codon. And there's also tryptophan, which also has only one codon. Something that's going to be super important when we get into control of gene expression in eukaryotes. We'll revisit tryptophan then. There are also 3 stop codons. Oh, sure, Benny. Just remind me before we finish so I don't forget, okay? So whenever the ribosome encounters one of these, it stops translation, right? So these are also super important, because you also don't want to just keep translating an mRNA to the very end, because it'd just be a waste, right? So every other amino acid is encoded by 2 or more different codons. And so there we go. Essentially, that's the process of translation, right? The mRNA is fed into the ribosome. We have our universal start codon, right, which is always a methionine. So that's an AUG, right? We have our tRNAs. There you go. It's supposed to be an A, right? And they're essentially the intermediate. Those are what translate those 3-word codons into this amino acid sequence growing in this direction here, right? And that's really the basics of translation. There's obviously an awful lot more to it than that, but it's kind of like the basics of that. All right, not an awful lot left to cover today. You'll be pleased to know, so you can go on with the rest of your day. Hour and 15 minutes is a bit of a heavy drag, so good on you for sticking with me. It's a lot easier when I'm drawing cats. So one of the really cool things about, so everybody goes on and on about DNA, right? Because it's the store of all information and stuff. But DNA is actually relatively boring. RNAs are super, super interesting. And they're interesting a lot of different ways. One is that they can form like really funky shapes, right? So what you're looking at here is a kind of stick and something model of, I'm pretty sure that this is, oh, I don't know what this is actually. I thought it was a tRNA, but it can't be because it's got a catalytic pocket. tRNAs don't catalyze anything. This may be a ribosomal RNA, it may be something else. But what you can see is that it forms all kinds of structures, right? So we have, we have a loop here, right? That's actually super important in tRNAs. We have some stems, right? This is where it's double-stranded RNA. We have all kinds of other funky things like catalytic pockets, if it's a ribozyme. That's also a fun word. Right, so it actually acts as an enzyme, which is super interesting. In fact, there's a real strong argument that the very first steps in the evolution of life were actually RNA. So RNA has the unique property of both being able to contain information, right, like an mRNA, and also to be able to catalyze reactions. And so they've actually been able to make RNAs that can synthesize themselves. I don't think they've managed it completely, but like 80% of itself, let's say. Right, so that's called the RNA world hypothesis, right, which is one of, not the only theories on the emergence of life, right? Because life had to start somewhere. It didn't start with a cell, right? That's somewhere, you know, kind of further along the process. It has to start with something. What that is, who knows? Right? Maybe it started with some other form of nucleic acid. Anyway, RNAs do all kinds of stuff. They're super, 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 super cool, in my opinion. They're also a complete total pain in the ass to work with as well, because you so much look at them funny and they disappear. And then we just come back to mutations. We've already kind of really covered these. One thing that I want to point out though, and this is a bit of a kind of a misconception, or at least a limitation in understanding, I guess, is that if you did generate a mutation in your germline and pass it on to one of your offspring, right, whatever mutation that was, whatever it was caused, there would only be one version, one copy of that mutation in existence in the whole population, right? So if you think about evolution as something that happens over huge periods of time which we really can't easily comprehend, not even talking hundreds of years, thousands maybe if the selection pressure is strong enough, but really we're talking tens to hundreds of, not millions of years, tens to hundreds of thousands, right? Is that one copy of that mutation, if that doesn't get passed on to your offspring's offspring, dies out, disappears, winks out from existence. So if you think about mutations, they are the sole source of genetic variation. There is no real other, I mean, I guess epigenetic changes maybe, but really most of it's mutation. That has to occur over many, 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 many generations before you can even think of getting to changes in the population, right? And so this is a kind of a chicken scratchings from Dr. Page start this course. Basically, you have changes, but then you have to select for those changes, right? Otherwise, those changes largely just disappear, right? Not through any kind of cleansing motion, right? Because that is also selection, just through drift, right? And I'll give you a cool example in the last couple of minutes of the class. So there's this kind of curiosity, right? Which is basically in Europe, there are a large number of people who are resistant to HIV, right? And they're resistant to HIV because they have one or two copies of a mutation which knocks out their CCR5 receptor, right? And without that, HIV can't get into CD4 plus cells and it can't replicate and cause AIDS, right? So the weird thing is, HIV has only really been in existence for 50 years or thereabouts, right? Emerged, they think, in monkeys in Africa, right? In the 60s to 70s. So how can 10% of the population in Europe have this allele if HIV, which selects for it, only came about 50 years ago or so? And so basically what they figured out is because of smallpox. So if you have a mutation in this receptor, they think you're more resistant to smallpox. And smallpox, over the last about 1,000 years of our history, basically killed a quarter of the population of the planet, right? Kind of hard to get that round your head, but literally billions of people have died from smallpox until it was eradicated, I don't know, 60 years or so ago, right? And so that selection pressure, basically smallpox kills kids, right? And if you child dies, it never reproduces, right? So if you have a mutation that allows you to survive to reproductive age, you're much more likely to pass on to your offspring. They're much more likely to survive to reproductive age, and so on and so forth. So anyway, it's a real fun story. It's kind of neat to look into. All right, that's enough of me chatting along and my absolutely terrible finger cam. So I'll post all this stuff up on Blackboard, and I'll see you in person next Tuesday. So no more Zoom. Woohoo! Actually, unfortunately, no more cute sleeping dogs as well. So you'll just have me to look at, unfortunately. Oh, well. So anyway, see you all next week. Have a lovely weekend and take care.
Can you also post the link from the other day?
Oh,
because
yes.
When you sent the email yesterday, the link wasn't there. So I just didn't. Not for me, at least.
Yeah, that's super weird because when I looked at it too, let me go back to Blackboard. We're the 05 section, I do believe. Hey, that's so strange. That happened to the other class too. Huh. Well, I'll try uploading the video in Blackboard itself. Given I only have a couple of them to do, then it should not screw up my storage limits. Yeah, I'm looking at the announcements. And it's disappeared. It was literally there. I swear I saw it. Little box with me in the middle of it. I'll upload it to Blackboard and let you know when I've done that, just so you can go and check it out. It'll be in the course content page in the first section. Thanks for the heads up, though. All right. Awesome sauce. See you all next week.